guys, Dr. Gabrielle Fundero here, RP Nutrition Coach, and today I'm going to introduce you to the gut microbiome. Now, according to Forbes, Google Trends analysis showed a 350% increase in searches for best foods for gut health from 2012 to 2017. Now, just about a year later, it's still a really hot topic, and most people are probably familiar with all the buzzwords that go along with gut health, gut microbiome, dysbiosis, and leaky gut. Now, this actually it's not an entirely new topic. People have been researching the gut microbiome for over a decade now. One of the biggest projects was the Human Microbiome Project and its counterpart, the Integrative Microbiome Project. This started in 2012 and lasted all the way until 2016. It received almost $200 million in NIH funding and came up with almost 200 peer-reviewed articles. And because they developed a reference base using this data, even more articles have been published based on this data since then. So the purpose was to understand the role of the microbial communities that cover our bodies and human health and disease. Humans actually have multiple biomes. We have an oral biome, we have a skin biome, we have a gastrointestinal or gut microbiome, we have a fecal biome, and women also have a vaginal biome. Now, all of these look pretty different, and it just so happens that the gut microbiome happens to be the largest and most diverse. Now, from these studies, researchers from multiple institutions were able to characterize about 99% of the microorganisms that inhabit these biomes. In the subsequent portion of that study, the integrative microbiome, researchers wanted to look at the influence of things like inflammatory bowel diseases, type 2 diabetes, and pregnancy and birth mode on the form and function of the microbiome. Now, in that Human Microbiome Project, they were able to take samples from multiple body sites from about 240 male and female volunteers, and that was sequenced as sort of the healthy core microbiome because they were healthy volunteers. So we have what appears to be a really good reference set of genetic information from these bacteria, and we can use that to compare to the biomes of other individuals. So what is the gut microbiome? The gut microbiome refers to the organisms, the microorganisms that inhabit your gastrointestinal tract. It's about 99% bacteria. The other 1% are archaea and viruses and perhaps even some parasites. There are so many microorganisms in there that if you were to collect them all in a bucket, it would actually weigh several pounds, which is pretty incredible. What's also incredible is that those 100 trillion or so bacteria are comprised of thousands of species expressing millions, perhaps, of genes. And so there's this idea that we were only about 1% human as we know it. If we were to count up all of the genetic information in our bodies, most of it would be from the biome. Most of it would be bacterial. These bacteria play an important role in energy harvesting from the diet. They synthesize nutrients and vitamins for us. These bacteria also play an important role in educating and maturing our immune system, even the pathogenic ones. They may even play a role in our mood and our behavior. Most of our serotonin is actually produced in the gut. So you really can't overstate the importance of the gut microbiome in human health and disease. So what are bacteria? These can be, like I said, pathogenic, that means they cause a disease, or they can be beneficial. They're single-celled organisms, they're relatively simple, and we can use their genetic information to identify that A, they're bacteria, and B, the actual specific type of bacteria that they are. We can classify bacteria by their shape. They may be rod-shaped, they may be spherical, they may be spirally, and then also their metabolism. Some of them are happy in an anaerobic environment. That means no oxygen. They're only going to be using glucose via glycolysis to produce ATP, and then others are what we say facultative anaerobes. So they can actually switch back and forth between using anaerobic and aerobic processes to make energy. Some of them produce gases. Some of them produce beneficial short-chain fatty acids. They can metabolize amino acids. They can metabolize glucose. So they're extremely diverse, just like any other organism on Earth. We usually, when we're analyzing a microbiome, we're going to analyze what genes are present there. We look at specific genetic material in these bacteria, and that can tell us that they're bacteria, because there are certain regions of this genetic bacteria that are conserved across all bacteria. But we can look at certain regions of that genetic information and from there determine the different species that are present. When we are classifying, when we're organizing that information into data we can then analyze, we usually will group them together in something called an operational taxonomic unit. Taxa is just a fancy word for saying group of organisms. So a taxonomic unit is a group of organisms. If you were to compare the genetic information between humans, you would find that all humans are about 99.99999% genetically similar. Compares to chimpanzees, 99.8888% genetically similar. Within an operational taxonomic unit, the cutoff for that is usually about 97%. So you can imagine that even though that sounds like a really high level of genetic similarity, there's still going to be some differences. There's still going to be some heterogeneity there. When we look at the influences that shape the microbiome, it looks like about 1/3 of our microbial species are going to be pretty much the same from human to human. It's just being human that gives us that microbiome. Just like dogs or mice or cats or birds will have different microbiomes, but all dogs will have some things in common. All birds will have some things in common. About 2/3 of our microbiome is individual. Now, we don't exactly know to what magnitude our lifestyles and dietary practices influence the shape of the microbiome, but it looks like perhaps each one, diet and exercise, may explain about 20% of the microbial diversity there. We organize these taxa by genetic similarity. So on this slide, you can see that we have something that sort of resembles a family tree in a really weird sense, but that's kind of what it is. So at the very top, at the kingdom level, that's the most distant relatives possible. So bacteria refers to a kingdom, just like Animalia refers to a kingdom. We can get a little bit more specific if we looked at the phylum level. But the phylum level is just like comparing animals with a spinal cord to animals that don't have a spinal cord. So it's still really, really general. Now, in most cases, when we're looking at research articles, they don't give us a lot of information about the class, order, or family level, although sometimes we do pare down to the family level. The best data that we can get would be at the genus or species level. Most people are familiar with Lactobacillus. Acidophilus is a specific species within the genus of Lactobacillus. It's one that we see on probiotics pretty commonly. So you can see that it's important that we get to that level, because just saying that we can see a difference in in animals with a spinal cord versus animals without, that's not very specific. If you're about to buy an animal, say off Craigslist, you're going to do a little Craigslist rescue, and they're only telling you the genus level, they're just saying it's a canis. Well, you don't know if that's going to be a jackal or a wolf or a dog. And you're going to need to know that, because you're going to have a very different parenting experience depending on what you're going to get. So from there, we want to know at the species level and even the subspecies level, because a wolf and a dog, still, that they're very closely related, but they're going to give you a different parenting experience as well. There are many ways to study the gut microbiome. Fortunately, we don't have to just culture an agar plate and stain it and see what shows up. Now we have advanced technology, and so we can actually look at the genetic information that's present in a given sample. In most cases, in human studies, we're looking at fecal samples. Now, I'll get into why that's not necessarily the most accurate representation in just a minute. It's just something to be aware of. Now, we can look at who's there and what they can do using metagenomics. So this is sort of looking at genetic blueprints or genetic recipe books for all of the different recipes we might be able to make. So you can say we have like a German cookbook, an Italian cookbook, a Russian cookbook. So we have all these different recipes that we could make. doesn't necessarily mean that we're actually going to make them. If we want to see what we're actually making, we have to look into metatranscriptomics and metaproteomics. So transcription and translation are the terms we use to describe taking that genetic blueprint, that genetic recipe, and actually making a protein from it. Metaproteomics measures protein content. So we can look at how much are we translating and transcribing these genes to make a protein, and then what are the proteins that we're making. So those proteins could be structural, they could be functional, could be enzyme. So we can actually look at the function of the bacteria in the gut. We can also use metabolomics. Metabolites are products that are either directly or indirectly produced when these bacteria are undergoing their metabolic processes. So metagenomics, who's there, what can they do? And then what are they actually doing? That's the functional side of the gut. So we can look at all of these and really get an idea of what's happening in terms of microbial diversity and then function. The gut microbiome spans the length of our digestive tract. Now remember, our oral microbiome is actually a little bit different. So when we're talking about the gut microbiome, we're really talking about the microorganisms that inhabit our stomach, small intestine, and large intestine. The stomach is really the main site of chemical and mechanical digestion of food. It's highly acidic. Acid controls microbial growth. So in the stomach, we have low levels of bacteria and also low levels of diversity. Really, the only bacteria that can hang out there are the ones that are okay in an acidic environment. In the small intestine, that's the main site of nutrient absorption for us. We don't see quite as many bacteria there because they aren't hanging out with the stuff that they really like to metabolize. But still, because we have lower levels of acidity and higher nutrient availability, our numbers of bacteria start to grow and we see increased diversity. The numbers and the diversity of bacteria in the gut really increase when we get to the colon or large intestine. That's the main site of fermentation. Because we can't readily break down dietary fibers, those pass to the colon pretty much unprocessed. Once they reach the colon, then bacteria can ferment them, make energy for themselves, make beneficial short-chain fatty acids for us. It's a very positive symbiotic relationship. There are a few things about the gut that influence the growth of these microbes. For one example, I mentioned acidity. Also, oxygen and nutrient availability. Some bacteria like to hang out in an anaerobic environment. Some of them can go either anaerobic or aerobic, so that means without oxygen or with oxygen. Nutrient availability, like I mentioned, we have more fiber available to the bacteria in the colon versus in the small intestine. Also, the structure of the bacteria and the structure of the gut play a role in what bacteria like to hang out where. If we take a cross-section of the intestine, we can see that it sort of resembles a hose, but with multiple layers. On the outside, we have a couple layers of smooth muscle. The smooth muscle produces wave-like contractions that we call peristalsis in order to move through food through the digestive tract. On the very inside, you see we have lumen. That's just a fancy word for basically the hole at the center of the water hose. We have a layer of mucosa there. That's a protective mucus lining that sits atop the intestinal cells. Now, intestinal cells do other things besides absorbing nutrients, but I'll just call them absorptive intestinal cells for now. Those intestinal cells are supported by connective tissue that has a lot of immune cells and immune tissue throughout. So you can think of this sort of like a poopy moat that is protecting the castle of our bodies, because that lumen is actually external to the body. And just like other tissues that come into contact with the external environment, it plays a very important immune function. So this is why we can sort of think of this like our first line of defense and why we have so many immune cells and immune receptors in the gut. If we look at the profile of the mucosal lining of that mucus there versus the lumen, we actually see that there are significant differences there as well. So the bacteria that hang out in the mucus layer tend to be more associated with protein metabolism versus those that are in the lumen. So remember, in the previous slide, I said that we're often using fecal samples to study the microbiome, but it's not necessarily the most accurate representation of what's going on. A fecal sample may closely resemble portions of the distal colon, so the part of your large intestine that's farthest away from your stomach. But as we move upwards towards the stomach, the fecal microbiome starts to look very dissimilar from the microbiome in the intestine. Likewise, like I said, if we're comparing the luminal bacteria to the mucosal bacteria, we're going to see significant differences there. So our fecal samples tend to be higher in bifidobacterium and lower in the bacteria that we'll see more proximally, like our lactobacillus that likes to hang out in the upper small intestine. So the Human Microbiome Project was able to utilize this advanced technology to determine what bacteria were present on the bodies and in the feces of those healthy volunteers. From there, they were able to identify that those microbiomes were dominated by 12 main phyla. But about 90% of all of the bacteria belonged to 4 phyla, and the majority of that 90% belonged to just 2. So it looks like our microbiomes really are dominated by Firmicutes and Bacteroidetes. And then we also have Actinobacteria and Proteobacteria. There are a handful of others, but they're extremely low levels. Diversity is a word that comes up a lot. We talk about gut diversity and how important it is, but what does it actually mean? Diversity refers to both the species richness and evenness within a sample or multiple samples. So species richness is the number of species that are found within a sample, while evenness is how evenly those species are represented. So if you take a look at this chart, you see that we can have a sample that has a very low number of species, a very low number of organisms, but they're all evenly distributed and represented. Or we can have a sample that has a lot of different species, but one's predominating. Ideally, we want to have a sample that's both rich and even. So we want to have a lot of bacteria present, but we want to have solid representation from our beneficial microbes and probably fewer of the pathogenic microbes. Not that we want to have zero, because those pathogenic microbes actually play a really important role in educating and maturing our immune system. We can also look at phylogeny. That's how closely related these species are. Just like if you're at a family function, you see aunts and uncles and cousins twice removed and brothers and sisters. We want to have that sort of, a bunch of distant relatives, not those that are all just closely related, because the more distant relatives that are there, the more diversity we have, the more functional diversity we have as well. Now, we can measure both alpha and beta diversity. Alpha is the diversity within one sample, whereas beta diversity is the diversity within 2 or more samples. So if we want to do a comparative analysis, we're usually going to be talking about beta diversity. People are often curious about how we even become colonized, and can we rebuild our microbiome, or how do we shape our microbiome? Well, like I mentioned in one of the earlier slides, it's probably pretty stable. Most of our microbiome, it probably is what it is, and we can't change it to a great extent. It's shaped very early in life. I mean, at birth. In utero, we're probably pretty sterile, and then it's at birth that we're first colonized. So the way that we're born can play a role in our colonization. If we're born vaginally, our gut microbiome will more closely resemble the vaginal microbiome of our mother. If we're born via a C-section, it'll more closely resemble her skin microbiome and perhaps also that of the environment around us. If we're breastfed, we'll get more of those skin microbes from our mother, versus if we're bottle fed, we may not be exposed to those. So bottle feeding in the past, especially in concert with a C-section birth, may have delayed colonization. But now formulas are starting to supplement with probiotic bacteria. So we don't see that there's quite as much of a delay because infants are still getting those beneficial bifidobacterium, for example. Now, as we reach the toddler years, that's when we start forming a microbiome that looks more like the way it will when we're adults and throughout life. This is in part due to an increase in complex carbohydrate and fiber intake in the diet. Now, breast milk still contains carbohydrates, but it doesn't contain a lot of fiber. So once we start incorporating fiber, that's when we see an increase in bacteria that can ferment that fiber. So for example, bifidobacterium. So that's a beneficial thing, and it's something to think about too, when we're starting to incorporate foods early in life, that can really make a difference. So we've seen in epidemiological studies examining the diets of children that those who were fed a westernized diet versus those who were fed a more agrarian, high carbohydrate, high fiber diet really had different looking microbiomes. And it seemed that the children who had a higher fiber diet had greater numbers of beneficial bacteria. So that's something to think about, that early in life, our dietary habits can really shape that microbiome. After those toddler years, things look like they stay relatively stable. Not to say that we can't have temporary derangements, especially if we go on antibiotics or if we have to use them chronically. That can certainly play a role in the diversity of the microbiome. Also, diet and exercise, as I'll talk about in subsequent videos, play a significant role in the shape of the microbiome. And also the development of obesity, which I'll talk about in later videos as well. Now, of course, diet, exercise, and obesity all kind of go hand in hand, because if you have an energy balanced diet and you're physically active, you won't develop obesity. But if you have a hypercaloric diet and or you are sedentary, then you're at greater risk of developing obesity. And those have all been associated with dysbiosis, or an unfavorable profile of gut bacteria. And I'll go into that a little bit more just in a minute. Now, as we get into older age, we see that there's a decrease in microbial diversity. This could be due to a multitude of reasons. Elderly individuals, especially those who are hospitalized for long periods of time, are at great risk of malnutrition. So it could be just poor intake of fruits and vegetables and fiber. It could also be associated with polypharmacy or taking multiple prescriptions at once. In any case, regardless of the cause, it's also another thing to be concerned about, because reduced microbial diversity reduces our immune function and reduces our capacity to fight off pathogens. One example would be C. diff, which is generally a hospital-acquired infection. It's a really virulent pathogen that can be life-threatening for individuals who are elderly. In most cases, we're exposed to pathogens all the time. Think about how many people probably touched the apple that you ate at lunch because you grabbed it at the grocery store. But if we have a healthy microbiome and we can fight those pathogens off, it's not problematic. We always need to have a balance of good and evil. So we need to have beneficial bacteria. We also need to have pathogenic bacteria. But if we don't have enough of those beneficial bacteria, we may not be able to control the growth of pathogenic bacteria. So what is dysbiosis? The Human Microbiome Project gave us a good reference of what could be considered a healthy biome, but there's actually no specific profile or definition for specific bacteria that indicate dysbiosis. And if anyone's trying to tell you to the contrary, that is not an evidence-based opinion. Dysbiosis is an umbrella term. Just like we have various forms of cancer, we can't have a cure for cancer because there's not just one form of cancer. We don't have a single form of dysbiosis, and we don't have a cure for dysbiosis. So dysbiosis takes many forms, and it has been associated with comorbidities or other disease states. It's a common characteristic of gastrointestinal disease states, as well as obesity and even type 2 diabetes. It generally refers to a lack of diversity. It could be reduced species richness or reduced species evenness, or even both. It's generally just an uneven representation of beneficial versus pathogenic bacteria, and it's associated with increased intestinal permeability and inflammation. Instead of intestinal permeability, a lot of people use the term leaky gut. Leaky gut was actually coined by researchers, but it's been a little bit used and abused. So I usually go with intestinal permeability. So what is intestinal permeability? Well, if you take a look at the picture on this slide, you can see that we have a healthy barrier where our intestinal cells are really nice and tight, close together, held together by tight junction proteins. And then we also have cells that have come apart a little bit. So the expression of these tight junction proteins has fallen. So levels of those proteins are lower. Now we don't have such great integrity of that cell wall. So you can think, if you're thinking of this like our moat, now the castle wall has started to degrade and villains and bad guys can slip through the cracks. So in this case, our villain, our bad guy, would be considered an endotoxin. So this is a toxin that actually comes from the bacteria that inhabit our guts. So in this case, the bad guy would be lipopolysaccharide, or LPS. It's an endotoxin that comes from the cell wall of specific bacteria. And when those bacteria break down, the LPS is then liberated. And then it can slip between these intestinal cells. At that point, it can interact with immune receptors or immune cells in the gut. One of these immune receptors is toll-like receptor 4. It's not just expressed in the gut, though. It's also expressed on skeletal muscle tissue. And we can actually induce insulin resistance in skeletal muscle cells by applying lipopolysaccharide. This has been replicated in cell culture studies and in mice, and we also see that there's an association between lipopolysaccharide, or LPS, and inflammation in humans. If this is chronically leaking out of the gut, we actually can see that too. We call that metabolic endotoxemia. Now, we can actually induce this acutely in a human by giving them a really high-fat meal and then measuring plasma endotoxin levels increase over time. We can also induce it in humans by giving them a really intense, extreme bout of endurance exercise in the heat. So that's not to say that the presence of LPS is indicative of a chronic inflammation or chronic leaky gut, because we can do it acutely, but it has been implicated in the development of insulin resistance and type 2 diabetes and is associated with both obesity and a high-fat diet. So what does dysbiosis look like? Well, it depends on what disease state we're talking about. In colorectal cancer, we see that there's an increase in harmful microbes. In inflammatory bowel diseases, it's a loss of beneficial microbes. In obesity, there's no clear-cut trend. So, like I said, dysbiosis takes many forms because there's no single form of dysbiosis. There's not necessarily a cure for dysbiosis. We're not exactly sure what causes it, but we know that it's correlated with specific disease states. Animal models can bring us to the closest possible cause-effect relationship that we can find in research. In one of the foundational studies of gut microbiome research, researchers found that they could actually induce obesity in lean mice through a fecal transplant. In other words, they made one group of mice obese. They then took the bacteria from those mice and transplanted them into mice who were germ-free, which means they didn't have any gut bacteria. Those mice then became obese through one of two mechanisms. They either ate more, no surprise, you eat more, you become obese, or they increase energy harvesting from the diet without necessarily needing to eat more. So this is really striking because it shows that the gut microbiome plays a role in extracting energy from the diet. And in fact, germ-free mice who don't have any gut bacteria, need to eat about 30% more to account for the lack of gut bacteria and just maintain normal body weight. So, we found the solution. Just knock everybody's gut bacteria out, we'll all be lean. Solution to obesity. But it's really not that easy because if we don't have any gut bacteria, that's actually really problematic and would probably lead to death because we need them to help extract energy from the diet, to help produce nutrients, to fight off pathogens. In mice that have lived without any gut bacteria at all, the protective mucus layer just breaks down so rapidly, and then they're completely open to infection by pathogens. So unfortunately, that's not that easy. But it's really a great method for being able to study the role of the microbiome in a bunch of disease states, obesity, and type 2 diabetes. So we can take germ-free mice and transplant them with the microbiome of other mice or humans. We can also have gnotobiotic mice who are transplanted with perhaps a single beneficial strain or a selection of beneficial strains or human bacteria. And then we have our conventionalized mice. So those are mice that grow up just like normal mice with a full set of gut bacteria. So these are all different ways that we can use a rodent model to study the microbiome and approach something that could be considered close to a cause and effect relationship between a dysbiosis and disease states. In humans, it's not so easy. We really are kind of stuck drawing correlations, and some of them really aren't that strong, but this is still an emerging area of research. Initially, we had some strong theories about the phyla level of bacteria. So remember, we talked about how that's very general. It's like looking at animals with a spinal cord versus animals without a spinal cord. So it doesn't tell us a lot about what's happening at the species level or the functional level. But this gave rise to many more theories and really opened our eyes to the importance of the gut microbiome in developing obesity, metabolic diseases, and associated comorbidities. Now, early studies, as I mentioned before, they looked at comparing a westernized diet that's high in saturated fat and refined carbohydrates to a more agrarian diet that was higher in complex carbohydrates and fiber. We've also been able to look at twins that were heterozygous for obesity. That means one twin was lean and one twin was obese. And we could study their microbiomes as well because they're the most genetically similar samples that we could get out of human beings. So early on, the idea was that perhaps too many Firmicutes could have been the problem and not enough Bacteroidetes. Firmicutes were associated with increased energy harvesting from the diet. They could ferment fibers to short-chain fatty acids and increase energy from the diet by anywhere from about 6 to 15%. That can really add up. Bacteroidetes were associated with leanness. Well, that theory doesn't really hold up because, as I'll talk about in some subsequent videos, actually, the production of short-chain fatty acids is associated with a lean phenotype, with lean body mass. So that's one theory that doesn't entirely hold up. Another theory is that perhaps a fiber deficiency could be part of the problem. If we don't have sufficient fiber, that means that we don't have a production of short-chain fatty acids like butyrate. Butyrate is really important for improving insulin sensitivity, for maintaining tight junctions, and thereby reducing possible inflammation via LPS. It can also play a role in the production of peptides that help to control appetite. So in that case, that theory does hold up pretty well, that perhaps fiber deficiency leads to increased intestinal permeability, decreased appetite regulation, so we're eating more, that leads to obesity, and then we have sort of a cycle that occurs there. Even though we don't have a chicken and egg, we don't know the cause and effect yet, just knowing that these things all tend to occur together is important, and that's something that we might want to avoid. Whereas in our agrarian diet that was higher in fiber and higher in complex carbohydrates, we had higher levels of beneficial bacteria like bifidobacterium that also, interestingly, produce these short-chain fatty acids that have been sort of vilified initially. So we don't have a very clear picture yet of who's causing what problem. And it's important that we don't limit ourselves to that we keep looking at the species level and at the functional level. Because when we say that, Firmicutes are causing obesity, that's sort of like saying vertebrates cause pollution. There's truth to that statement, but it's much more nuanced than that. There are so many vertebrates on this planet. If we really want to work on improving the ecosystem and benefiting the environment, we really should probably figure out which of those invertebrates are causing most of the pollution and look there. So I hope you guys enjoyed this video introducing you to the gut microbiome and the preliminary research in the field. I have other videos that are available to you if you are interested in learning about the relationship between the microbiome and diet, or the microbiome and exercise, and even some practical take-home tips and some fecal fallacies that you want to watch out for. This is still an emerging body of research. We have a lot to learn. We've got more questions than we have answers right now, but I hope you guys enjoyed diving into the literature a little bit there and come back for more.